Perfect. All right. So welcome everybody to our VacCAP webinar series. My name is Amaya Tucha. I'm part of the extension team at VacCAP, and I will be moderated for today's webinar. We have with us today Hiduk O that will be discussing uncovering the genetic control of organic acids and sugars in blueberries. So we're very excited to have Hiduk with us today telling us about his research. Hiduk has a bachelor's in science and an MS in horticultural science from the Seoul National University in South Korea. He is currently a PhD student at North Carolina State University, working with Dr. Massimo Irizzo and Dr. Penelope Perkins VC. His dissertation project aims to understand the genetics underlying quality characteristics in blueberry, such as taste and texture, and also understand the key players determining fruit shelf life in blueberries. So thank you, Hiduk, for being a speaker today and presenting some of your research to the people that are attending the webinar. I want to remind everybody to please stay muted during the talk. And if you have questions, to just you can put them in the Q&A or in the chat. And we will be recording this webinar and posting it in our YouTube channel if you want to go back and review some of that. And so with that, I'll just let Hiduk take over and start his webinar. Thank you.
Okay, can you hear me okay? Okay.
Perfect.
Thank you so much for the introduction. And thank you for having me here today and giving me the opportunity to present our work here. Hello, everyone. Thank you very much for being here today. My name is Hiduk O, and today I'll be sharing some very interesting results that we've been getting from our research to understand the genetics underlying organic acid and sugar contents in blueberry. So I work with blueberries, and like a lot of you that are here today do too. And one thing I really enjoy about working with blueberries is that when I meet new people, I say I'm a grad student and I work in the lab, and they're like, okay, that's cool. And then I say I work with blueberries, and they're like, wow, that's amazing. I love blueberries. So evidently, everyone loves blueberries, and data shows that too. The consumption of blueberries has increased rapidly during the past few decades because they taste good, and they have a lot of health benefits too. The production of blueberries in the US has increased almost fourfold since the year 2000. Product availability was the major factor leading that market growth. Originally, the cultivation of high-bush blueberries was restricted to temperate climates with long chilling hours, but after successful breeding efforts, now many more cultivars are available that can be grown in warmer environments in the South with less or no chilling hours. After years of expansion in growth areas and product availability, now the consumers and industry are more selective for fruit qualities such as taste and flavor, consistent texture, shelf life, appearance, and firmness. The issue was, even though the breeders were always thinking about fruit quality, they didn't have much actual empirical data to use for selection. For example, there wasn't much information telling you, okay, this is what determines blueberry taste, and these are the genes responsible for it, so you can use this for your breeding. So part of the VacCAP project was find the links between fruit characteristics and fruit quality and the genetics. We want to figure out how fruit characteristics such as appearance traits, texture, and chemical composition are related to fruit quality that decides consumer preferences, and trace it back to the genetics, looking for QTLs and candidate genes so that we can develop DNA markers for breeding programs. Out of the fruit characteristics, my talk today will focus on non-volatile chemical compounds related to taste and flavor, and the related traits such as pH, titratable acidity, total soluble solids, organic acid content, and sugar content. I'm going to first lay out the connection between these fruit characteristics and fruit quality, and then I'll share our work so far on connecting these fruit characteristics to the genetics of blueberry. When we think about what the consumers care about in terms of fruit quality, when it comes to taste, the consumer's liking has been related to sweetness and sourness. The overall liking of blueberries increased as they were sweeter and decreased as they were more sour. This quality preference regarding sweetness and sourness is directly related to total soluble solids and titratable acidity. Titratable acidity is positively correlated with sourness intensity in blueberry and negatively correlated with sweetness intensity. And on the other hand, total soluble solids was positively correlated with sweetness intensity. And this is related to the overall liking as well. Total soluble solids positively correlated with the consumer's liking, and titratable acidity was negatively correlated. And when we go a little deeper into the chemical composition of the fruit, the sweetness, the sourness, total soluble solids, and titratable acidity are all closely related to the contents of sugars and organic acids in the berry. So now we have some idea on how fruit characteristics such as total soluble solids, titratable acidity, organic acids, and sugars are related to fruit quality. But the link to the genetics is still not well understood. Understanding the link to the genetics can be crucial to breeding programs because identifying putative candidate genes and developing molecular markers can facilitate marker-assisted breeding, which can help reduce cost and resources that is needed for breeding and accelerate the breeding cycles. So as part of the project, working towards this goal, we have started to identify some QTLs related to pH and titratable acidity. Dr. Felipe Ferrao in Florida identified SNPs associated with pH, and Dr. Mola Mengist from North Carolina found stable QTLs on chromosome 3 in 2 different biparental populations, Reveille by Arlen and Draper by Joel. This means that this genomic region on chromosome 3, that is boxed in red here, has been repeatedly proven over years and across populations to be associated with pH and titratable acidity. Since organic acids and sugars are closely related to these traits, we continue these efforts and phenotype the F1 biparental population, Reveille by Arlen, or R by A, for organic acids such as citric, quinic, shikimic, and malic acids, and sugars such as fructose, glucose, and sucrose, on top of pH, titratable acidity, and total soluble solids. So this is an extended effort to further understand the links between these fruit characteristics and the genetics of blueberry. So looking into the phenotypic data that we collected from the 346 genotypes in this population, extensive variation was observed for all traits. pH ranged from 3 to 4. Titratable acidity mostly between 0 and 1. Total soluble solids between 10 and 15. For the organic acids, the main organic acid in this population was citric acid, followed by much lower contents of quinic, malic, and shikimic acid. And for sugars, the main sugars were fructose and glucose. It was nice to see that the variation of these traits was consistent across the 2 years of data collection. The red histograms were 2021 data, and the blue was 2022. And the distribution largely overlapped between the 2 years. But this was a biparental population, segregating from 2 parents that all the offspring shared. So we were curious what these variations were like compared to other blueberry genotypes outside of this population. So we compared the R by A data that you see on the top row to a data set from a diverse list of cultivars that we have here in North Carolina. which is what you see on the bottom row. The diverse set of cultivars that we have here consists of 61 cultivars. That includes northern highbush, southern highbush, and rabbit-eye cultivars. So it's a pretty wide range of genotypes. And if you compare the variation of each trait between these two populations, the variation in the R/A population was pretty comparable to the variation we observed in the 61 cultivar population. So what this tells us is that we got a very nice variation in our data from the R/A population to work on the downstream analyses. And to get to know our data a little better, we looked at the correlations between these fruit characteristics. The first thing I want to point out is that citric acid was highly correlated with total organic acid content and pH and titrative acidity. The correlations with titrative acidity and total organic acid content were highly positive, and the correlation with pH was highly negative. This indicates that citric acid, which is the predominant organic acid in this population, explains most of the phenotypic variation of titrative acidity and pH. If you look at the sugars, they were positively correlated with each other and also with the total soluble solids. However, sucrose, which was the minor sugar in this population, wasn't correlated with total soluble solids and had significantly weaker correlations with the other sugars. So this suggests that fructose and glucose are the major players in this population for total soluble solids. So we have data collect across 2 years. So we also looked at the broad sense heritability of these traits. If we look at the traits related to acidity, like pH, titrative acidity, and the organic acids, the heritability was relatively high, except for malic acid. The heritability was higher than 0.7 for these traits, which means that most of the phenotypic variation was due to the genetic variation. However, in the cases of total soluble solids and the sugars, the heritability was mostly lower than 0.5, which means that more than half of the phenotypic variation was due to other factors like the environment. Using this phenotypic data, we were able to detect QTLs for pH, titrative acidity, and organic acids, which was stable across multiple years. If you're not familiar with these plots, they are a way of showing the results of a QTL analysis. The x-axis is the physical position of a chromosome. So this is showing chromosome 3, just one chromosome. And chromosome 3 is about 120 centimorgans long in the linkage map constructed here. So on the bottom, you can see that the x-axis spans from 0 to 120. The y-axis in the line graphs is the LOD score. It indicates how strong each position of the chromosome is associated with the trait. So a higher LOD score means that that part of the chromosome is strongly related to that trait. And the dashed line going through the middle is the threshold for significance. So these plots are first showing you the QTLs detected on chromosome 3. If you recall that previous studies have found QTLs for pH and titrative acidity on chromosome 3, those QTLs were detected again in the same region consistently in our study, which is the plot on the left. And in addition to that, we also identified QTLs for total organic acid and citric acid contents on the same chromosome region, which is the plot on the right. As we saw earlier, pH and titrative acidity are important traits affecting the taste, and citric acid is a key organic acid related to pH and titrative acidity. So the location of this QTL is a very important genomic region to search for candidate genes and potentially develop markers. We found some very strong QTLs for other organic acids as well. A major effect QTL for quinic acid was detected on chromosome 4, and a QTL for shikimic acid was detected on chromosome 5. They were both stable over the 2 years. So now we have a pretty good idea on which chromosome and which part of the chromosome is related to these 3 organic acids, citric, quinic, and shikimic acid. We narrowed it down to specific regions on each chromosome. However, these regions that we identified here are still pretty big portions of the chromosomes. These regions can contain hundreds of genes. So this is a very good start, but we need to dig deeper into these regions and find out which genes are responsible for controlling the content of these organic acids. Out of the hundreds of genes that could be in each region, which ones are actually responsible? One way to look for these possible candidate genes is to select a few individuals that have very distinct genotypes and phenotypes and conduct an RNA-seq experiment. How it works is we'll have very distinct individuals, and assuming that the differences between those individuals are from their genetics, we'll extract all the RNA from these individuals and compare which genes are expressed differently between them. This way, we'll be able to pinpoint the genes that are responsible for altering the organic acid contents. The way we selected the individuals to do this in the study is we first looked at the QTL analysis results, and within the QTL region, we grabbed a few markers that are likely contributing to the trait, and we checked the allele dosages for these markers in each of the individuals in the population. This way, we can find individuals with high allele dosages of these markers and those with low dosages. This is showing you an example of how we did it in quinic acid. On the heat map here, the first 3 rows are the markers we chose for quinic acid. They are within the QTL region, and the different colors indicate the allele dosages for different genotypes. The dark navy indicates 0, and yellow indicates 3, so it goes from 0, 1, 2, and 3 on this heat map. So ideally, we want to choose individuals that have high dosage for these markers and some that have low dosage. Next, we look at the phenotype. In this example, we're looking at the quinic acid content of each individual or genotype in 2021 and 2022. So these 2 bottom rows are the phenotypic data for quinic acid in each year. And the brighter colors in green indicates higher quinic acid levels, and dark navy indicate low quinic acid levels. So overall, this gives us a good visual on which genotypes have high or low allele dosages and which ones have high or low quinic acid contents based on the 2-year data. Now we select 3 genotypes on each end, 3 from this end and 3 from the other end, which will be the 3 biological replicates on each side. And then we extract the RNA from these individuals, sequence them, and see which genes are differentially expressed. And hopefully we'll be able to put our fingers on some genes that we can say, okay, these genes are responsible for the variation in quinic acid. That was an example of how we selected genotypes for quinic acid, but we did the same exercise for citric acid and shikimic acid too. We selected 6 genotypes for citric acid, 6 for quinic, and 6 for shikimic, and sampled them this year for RNA extraction. Just to make sure that they have distinct phenotypes, we quantified the organic acid levels in these samples and confirmed that they have very different organic acid contents. So we went ahead and extracted the RNA and sequenced them. And I'm currently working on the RNA-seq data that we got back to identify the possible candidate genes for these organic acids. We're searching within the QTL region. which genes are differentially expressed, and which of them could be the candidate genes for these traits. And we're expecting to have some answers very soon. Another part of our research that I wanted to briefly share with you is the post-harvest side of the story. The genetics we were looking into was at the time of harvest. However, most consumers don't get to eat the berries right at harvest time. It takes some time for the harvested berries to reach the consumers. In some cases, this could be weeks. So we were curious about how all this is related to the storage side. The variability of these traits during storage is also unclear. How would the genetics of these traits carry over to the shelf life? So we looked into how stable these traits are during storage. We stored the berries from a large set of cultivars. The one that we got a glimpse at earlier that has 61 cultivars. We stored the berries of these 61 cultivars at 2 to 3C and 80% relative humidity over 6 weeks, and phenotype these traits to see the variability during storage. We phenotyped the samples at 0, 2, 4, and 6 weeks of storage. Long story short, the changes of these characteristics were relatively minor. Statistically speaking, we did find some significant changes over the weeks of storage, but realistically, they seem to be rather minor changes. We looked at the individual cultivars to get a closer look. For example, these are the 61 cultivars for pH on the left, and these are the 61 cultivars for titratable acidity on the right at 0, 2, 4, and 6 weeks after storage. And what you can see is that the changes over storage are relatively minor, and the degree of changes are mostly cultivar dependent. So what this means is that the chemistry at the time of harvest will pretty much carry on to the shelf life. To wrap up, here's a summary of what I talked about today. To understand the genetics of fruit characteristics related to taste, we phenotyped a biparental population, Reveille by Arlen, for pH, titratable acidity, total soluble solids, organic acids, and sugars. Extensive phenotypic variation was observed for all traits in this population. The variation was even comparable to a diverse set of cultivars with a very wide range of genotypes. Out of the organic acids, citric acid explained most of the phenotypic variation of pH and titratable acidity. Moderate to high heritability was observed for pH, titratable acidity, and organic acids. And the heritability was much lower for total soluble solids and the sugars. Major effect QTLs that were stable across years were identified for pH, titratable acidity, and organic acids, whereas no QTL was detected for sugars in this population. We are currently working on identifying candidate genes within the QTLs. And in terms of post-harvest storage, pH, titratable acidity, total soluble solid, and contents of organic acids and sugars were mostly retained throughout shelf life. So with that, I want to thank everyone who are hosting this webinar and giving me the opportunity to talk about my research. I also have people to acknowledge and thank who are helping me navigate through my PhD research, including my advisors, Dr. Penny Perkins-Vesey, Dr. Massimo Rizzo, who are, the best mentors and teachers. All the people in both labs that helped me handle the samples, phenotype them, and analyze the data. Dr. Ashley Yao, who now is a postdoc in Dr. Holzkamp's lab, Dr. Marcello Molinari, Dr. Suzanne Johanningsmeyer, and everyone at the Castle Hayne Research Station. I simply couldn't have made it this far without these people. And of course, I want to acknowledge NIFA funding the VacCAP project. Thank you so much. and I'd be happy to take any questions.
Great. Thank you, Hiruk, for your presentation. Do we have any questions? And people can, paste them on the chat, type them on the chat, or if you want to just ask a question directly, you can also unmute yourself. We have one question in the chat. I can read it for you, Hiruk, from Jessica. Do you expect to select for or against shikimic acid content once you find a marker?
That's a good question. At least in highbush, it seems like citric acid is the major organic acid, and it's known to contribute to the sourness. It's very highly correlated with pH and titration acidity. So I think that would be a key organic acid to select. For, I'm not too sure about shikimic acid. We do get a strong signal for QTL, so we are looking into it with the others, but I'm not sure how it affects the taste of blueberry, how it affects the flavor, and how important it is to select for. Yeah, please educate me if you know something about this.
Great. So I see that Janine has raised her hand. So Janine, if you want to unmute yourself and ask the question, go ahead.
The question's actually from Fernando, but I might have a question after too.
Yeah. Hey, Hiruk, great presentation. I had a question about why you decided to map all of the different acids individually. And do you have an indication of whether taste is a function of multiple acid types rather than just a single acid type? Thanks.
Yeah, that's a good question. Well, pH and titration acidity are obvious and easy to phenotype characteristics of the fruit that directly affect the taste. So that's definitely was included. And there were some previous studies on that too. But organic acids are, as far as I know, are the main chemical components that control the pH and titration acidity and acidity of the berry. So that's why we wanted to look at the organic acids, how they all function together to decide the overall taste or flavor. I don't think we have a very good picture on that. Based on previous studies, citric acid seems to be contributing to how sour it is. And there are some studies on quinic acid contributing to the bitterness or astringency, but I'm not sure how positive that is, how confident that is in blueberries, in the case of blueberries. So I think there is some interesting future work to do to figure out how all these organic acids contribute to the final taste or flavor. But just based on previous knowledge and literature and all that, we know that organic acids do contribute and play a major role. So we decided to look at them individually to see if we can point them out.
Hey, great. Thank you.
Hi, Hiruk. My name is Janine. I have a question. I really enjoyed your talk, by the way. I wanted to ask about your graphs where you showed that pH and TA and TSS, they don't change over time. So it's kind of like a post-harvest study. Which population was that tested on? Was that on the same biparental population that you did the QTL mapping?
That was on a set of cultivars that we have here in North Carolina. It has 60 or so cultivars, includes northern, southern highbush, and rabbit eyes. Yeah, so it's a mix of a lot of different cultivars. Yeah, it's not the biparental population, yeah.
Thank you. And would you expect those, would you expect that same stability in different production environments?
That is a good question. I don't think I have any data to answer that question. It would be very interesting to see if it does. We only tested one storage condition too. So yeah, it would be interesting to see it different locations, different storage conditions too.
All right, thank you, Hiduk.
Thank you.
If anybody else have any other question, you can either raise your hand or just type it on the chat. I don't see any more questions on the chat, but if there's anybody else that would want to ask a question, just give. Okay, so Jessica asked, what was the highest amount of sucrose that you saw?
Sucrose. I don't want to give you false information, so I'll go back to the slides. Sucrose. Seems that, so this is dry weight, and it's milligram per gram, if I have it right. And it was 25, the highest was around 25 milligrams per gram dry weight.
And like any other questions? Give it a little bit longer to see if anybody's typing.
I mean, I think understanding how all this translates into the actual, sensorial experience that we get from eating the berries is very important. It's a very important piece of the puzzle. So, if anyone has any insight or to share about that, please shoot me an email or let me know. I'm very curious if there's anything I'm missing or I don't know about.
Well, it doesn't look like we have any more questions, so I'll just reiterate and put again in the chat that we will post a recording of this webinar on our YouTube channel, and if you want to go and look at some previous webinars, you can also find them there. I see that Hiduk also added his email on the chat if anybody has any more questions or some comments for him on his research. One last question, I guess, here from Janine and Fernando. Well,
hey, Hiduk, one more question. I might have missed it at the beginning of the talk, but when you for the biparental population, when each of your plants was giving you your fruit, how much did you reserve for your own individual tasting? Because that's something that in my own research I often think about. Sometimes each of the plants isn't producing a whole lot of fruit, so you want to mine the data from the fruit, but at the same time, like you just said, all of the individual components don't are you need to taste it rather than just like get all the information. So I was wondering, given that you just mentioned that about where it's hard to explain the taste from the sum of all these different parts. Did you get a chance to, for any of the individuals, did you get a chance to taste? And and then did you see anything in particularly interesting from that planet on its own in the data set?
And Hiduk, just to give you a perspective of where Fernando's coming from, he works on cranberry blueberry hybridization. So he's made crosses that we call cran blues.
That's very interesting.
Yeah.
Yeah, it's a whole other ball of wax. But yeah, go ahead, Hiduk.
Yeah, we have about 350 genotypes for this biparental population, and I did not taste all of the genotypes as we went along, but we did select a few, maybe 10 genotypes, and sampled them separately, and we did like a small fun sensory panel test here. We had some previous data on which ones had high touch acidity, which ones had low touch acidity. So we kind of picked the extreme highs and lows and saw if we could, tell the difference with our taste. And a lot of them we were actually able to say, okay, these are actually sour. We did a blind test on them, and a lot of them actually turned out to be, in the sensory way, it was very sour and it was bland. But there is a sugar component to it too, and the ratio can be important too. And that wasn't really differentiating any of the individual organic acids. So that could be future work too. And that was just, for our own knowledge. It was like just a small fun experiment that we did. Yeah, just to kind of confirm that the data to some extent agrees with the actual sensorial experience we get. Yeah.
Well, that sounds great. That's very, very valuable to do. But yeah, thank you so much for sharing.
Thank you.
We have one more question in the chat, actually two now. So the first one is the variation in the shikimate content somehow correlated with differences in color that you can visually appreciate?
We did not evaluate the color, so I don't have data on that. Is there a known relation between the shikimate acid and the colors? That's a very interesting viewpoint.
Yeah, maybe Monica can put in the chat a little bit more why she thinks those things are correlated. Maybe something that you didn't know about. There's another question that says, great presentation, just curious on your opinion, since consumers prefer sweeter blueberries, what would be the consequences, if any, of breeding organic acids out of blueberries?
Yeah, that's a good question. I mean, it's not just the sugars, or it's not just the organic acids that decide the taste, right? It's a contribution from both sides, and the ratio is important too. From what we're seeing, organic acids seem to be a big player in the overall taste, because based on the variation that we're seeing, the variation in sugars isn't as large as the variation of the organic acids that we're seeing. So the guess is probably the organic acids might play a bigger role. So I think it definitely is a potential very important trait to breed for. I'm not sure, like, if you actually do it, what would be the consequences. Definitely the change in taste would be one of them. I'm not sure about the other sides. I mean, they are, for example, citrate is directly in the TCA cycle. I don't know what would happen if you select against it. That's a very interesting question.
Great. So Monica says that shikimate is the precursor of phenolics and compounds given color. So that might be one of the reasons why just lower amounts of it might potentially influence color development.
Right. I mean, if it is related to color, then that would be a very interesting, component to explore as well.
Yeah.
Yeah, that's very interesting. Thank you.
And Janine has I think that, you answered these questions, that what would be the physiological consequences of breeding all of the organic acids out of the fruit? And as you said, I think that would have repercussions beyond just the taste.
Yeah, I mean, I'm not super familiar with, previous literature on this aspect, but a lot of these are substances that are involved in primary pathways. So I'm not sure how the actual consequences would turn out to be. I think that's something we should learn. Yes.
Well, I think that those are all the questions that we had. I just want to thank Hiduk for a great presentation and for answering all the questions. I also want to just let everybody know that there will be a survey that we will be sending you. So if you can please take a couple of minutes to answer it and provide us some feedback about how we can do better, we would really appreciate it. So with that, we're just going to finalize our webinar for today. Thank you, everybody, for attending, and thank you, Hiduk, for a great presentation.
Thank you, everyone. Thank you for the great questions. And, email me with any more questions, or if you have any comments or feedback, I would love that. Thank you so much for having me today.